So in this video, we will now be looking at the different methods at looking at the affinity of the protein ligand interaction, especially the lipid and membrane protein ligand interactions. So first, we're going to discuss the lipid interaction first, because it's a very hot topic right now. So protein lipid interaction is crucial because they are involved in disease and they're modulating ion channels activity. So PIP2 bind to KIR channel, for example, will activate the channel and opens the channel. Also, we have some work, but cholesterol binding to KIR channel would inhibits the channel. And so it's very interesting to find not just where lipid binds to an ion channel, but how strong does it bind? And there are several ways that we can looking at protein lipid interaction to the channel. So one of the technique is called PMF. So we have an ion channel protein, and we would have a lipid binding initially on a site here. And what we have here is we apply force to be steering the lipid away from the binding site physically, and we pull it away, and we take snapshot along these coordinates, and we sample the force along these paths. And so we sample the force along these paths. And as you can imagine, if it's close to the protein, these lipids are going to want to come and interact with the protein. So you have to apply stronger force to pull back and toggle it backwards so it doesn't interact, right? And we can measure the force required. And what this gives us is an energy landscape, a one-dimensional energy landscape, where it would tell us how strong it is. And of course, it will go towards zero, because as you pull it to the bilayer, it won't be interacting. And this will give us a relative binding energy of a lipid to an ion channel. And this method, as you're measuring the force, an average force at a certain point, and that's why we call it a potential of mean force calculation, potential of mean force, or PMF calculation. The other way, which is my favorite method, is that you can have an ion channel bind to one lipid, let's say in this case PIP2, and what we can do is we can turn PIP2 over chemical space to the other lipid. So you can go from one lipid to the other lipid species in hybrid space, right? So you have PIP2 turning into POP-C in a bound configuration, and you can turn PIP2 in just a bilayer to POP-C in a configuration. Now you might wonder, what does this actually means, right? So this case, what we are doing practically is calculating the energy as PIP2 sample on a protein and sample on a lipid. So this is what we would like to find out, right? This is delta G binding of PIP2 to the protein. And this is another case where we have PC, and we know that we have PC bind to the protein. Now I can tell you now, as a surprise guess, that if I run PMF on this, what I get is delta G of this binding event. I get a very near to zero, because PC is in bulk equilibrium, and we know that in equilibrium, you don't get much binding of a lipid. So this thing will exchange all the time and have very weak binding. So delta G is very close to zero. So what I can do is I can practically turn PIP2 to PC in alchemical space slowly as it binds to the protein in the bound configuration with the protein, and in the bulk lipid, so in just the bulk lipid bilayer, and then calculate delta G2 and delta G1. And what I can do here is take delta G2, subtract to delta G1, and what I have is the delta G bind of how the binding free energy I've got out of this process, which is somewhat slightly cheaper than the PMF method. But of course, we're losing the landscape of the energy that we can get in a one-dimensional axis here. So, method of choice, you can use two ways, or, one way or the others, to test the binding affinity of the lipid. So these are currently mainly being done at the coarse grain level, because liquid equilibration in a bilayer is very slow. But we are looking for the way that we can do this in atomistic as well, which can be very interesting in case we're looking at unsaturated versus saturated, or doing it with charged species, etc. So these are currently a very hot topic of study, and what everyone has been very interested in. In the next one, we will have a look at ligand or more complex drugs and how they might be important.